to our second speaker, Rahuman Sheriff, on reproducibility and systems biology modeling. Are you queued up and ready to go?
Yes, thanks.
Okay, I will turn it over to you.
So thank you for giving me a slot to speak today. So the title is slightly different. As you can see, it's the reproducibility in systems biology modeling and biomodels role. So in this talk, I'm going to talk first about biomodels, what it is about, and then about the study that's done in biomodels. Right. So what is biomodels? For those who don't know, biomodels is a database of mathematical models of biological processes. It was established about 15 years ago as a platform for modelers to exchange models with each other. Right. If you're a modeler, you can go to Biomodels. You can submit your model, which can be in any modeling formalism. It can be ODE, logic, Petrinet, any model, constraint-based model, and they can all be submitted in to Biomodels. And these models can be in any format. They can be a MATLAB script or a C or Python script or standard such as SBML, SLML, and so on. And these models, upon this submission to Biomodels, are searchable and can be found by other modelers. So one advantage of submitting a model to Biomodels is the curation. So models submitted to Biomodels are carefully checked by the curators in my team. So by the way, I lead Biomodels team, and they compare the equations in the publication of that manuscript, the manuscript describing the model, and they encode the equations in computer readable standard formats such as SBML. They run simulations using open source tools and try to reproduce the simulation results. If a model is reproducible, if it does what it's supposed to do, and if the simulation matches what is described in the publication, the simulation figure in the publication, then we stamp this model as curated and reproducible. This is very good because then the users who come to Biomodels can look for models which are verified. And it's also good for the modeler who submitted the model because there is some external verification or validation of this model. And we have seen that users tend to download these verified or curated models twice more than the non-curated models. Okay, so it's not just the models from the curators, but we also do targeted curation. We try to find certain area of research and then try to curate more and more models and add it to the Biomodels database so that modelers can benefit from that. Currently, we are focusing on curation of COVID-19 models. We have a good collection of COVID-19 models, and if you can go to the link shown below in this page, you will have a good collection. All these models are open source and it's free to use, so anyone can download them and reuse them. Okay, and we have many more collections. So if you go to Biomodels, you can search your keyword of interest, immune response or COVID-19. You'll find a list of models, and then you can filter them based on the facets on the left-hand side, and narrow down your search and find the model of your interest. Once you go to the model, you can see a title of the model and then the manuscript. You can go to you can click the link and go to the publication, or you can also find some quick overview of this model. What is it about? Which organism it is. Like on the right-hand side, you see some metadata saying that it's for humans, Homo sapiens, and it's about T-cell regulation. So we add extra ontologies and control vocabularies to make these models more semantically enriched, and so they are findable by search. And also gives meaningful information for the modelers who try to reuse or repurpose these models. Okay, moving on. Biomodels is widely used. In a survey of scientists, many of them said that they go to Biomodels because it's one of the important data source for them. And also many modelers prefer to submit their model to Biomodels after supplementary material, GitHub, and personal website. As I told you, this is again because of the curation services probably, and also the models are more findable when it's in such a repository. Okay, so you can read more about our work here from these publications. I will move on to the next slide quickly. Okay, now coming to the main topic of the discussion. Reproducibility in systems biology modeling. So before we go there, we need to understand the terminologies so that we are on the same page. So replicability or repeatability is the ability to use the same code provided with the manuscript using the same software to reproduce the simulation results. Yeah, but reproducibility is the ability to recreate model without original code. So replicability or repeatability is using the same code, using the same software, reproducing the simulation. But reproducibility is the ability to recreate the code without recreate the model without the original code. So in our study, we focused on the latter, that is the reproducibility of the model. So we assessed reproducibility of 455 models from published peer-reviewed articles and tried to find how well they are reproducible. So this is one of the largest study ever done to assess the reproducibility in this field. Right. So what did we do? We encode the models in SBML, and we get the parameters values from paper, supplementary material, put them all or from the authors and put it together. So why do we use SBML? SBML is one of the standard community standards used because it's highly interoperable. If you have a model in SBML format, it can be imported into several softwares, over about 300 softwares. Tools support SBML. So if you can import it into MATLAB or Python or R or Java. So you have huge flexibility. So when you share your model in this sort of community standards, you can import them in several formats and reuse them. Okay, so we encode the model in SBML. We run the simulation using open source tools such as Copasi, and then we try to see if the figures are reproducible. So we consider a model as reproducible if at least one simulation figure from the publication could be reproduced. Then we are happy. We say this is reproducible. So we still we are not very strict. We accept minor deviation if it doesn't affect the scientific conclusion of that study. If a model is not reproducible, we try to fix it. Let's say there could be some error in the equation or parameter. We can try to play around and tweak the model and empirically try to fix the model. If we still can't fix it, then we try to contact the authors and get the information from the authors and try to fix the model. So now the results. Before that, give an example. Yeah. Sorry. So let's say you have publication with certain simulation figures for the model described in that manuscript. We reproduce it, and this is part of the curation which you do in Biomodels, and this figures are reproduced and added to the curation tab of that model in the Biomodels. So you can cross-verify that these models are indeed verified by someone and reproduced. Okay, so in this study, we have curated about 455 kinetic models, that is ordinary differential equation models of biological processes. These are from peer-reviewed research articles from 152 different journals published from 1980 to 2020. So this activity was done as a part of normal Biomodels curation, and hence the models are not randomly sampled, but they are selected based on the priorities of curation in Biomodels. So this 455 models are a significant proportion of models available in Biomodels. Okay, now the results. Sorry, now here is the main result. So we would expect that these mathematical equations are reproducible, but unfortunately, only half of the models are directly reproducible. Half of the models could not reproduce based on the information provided in the manuscript. Of course, this was of course surprising and also not, because we have heard from the community always that there are models which do not reproduce, but no one knew how much prevalent is this. And from our study, we see that at least half of the models are not reproducible directly based on the information provided in the manuscript. Okay, so those models which are not reproducible, we went and did put some additional effort. As I mentioned, we tried to do empirical correction. We tried to tweak the model, look for typos, any mistakes in the equations, or any typos in the parameters. It could be 0.1 instead of 0.001. We try to play around a bit and see if we can fix the model. Yeah, and by doing that, we almost fixed 9% of the models in total. And then we try to contact the authors, and from the authors' support, we kind of reproduce 3% of the model out of 455 models. And finally, 37% of the model could never be reproduced. Yeah, we could not reproduce based on information provided or from even additional effort. Right. So when we tried to contact the authors, it was not an easy task. Of course, there were publications which were very old. We could not even reach authors, but then we still managed to contact about 90 authors, and majority of them, 70% of them, do not respond. Yeah, even though it's good for them to have the model somewhere in a public domain, open for openly visible, they did not respond, and only 30% respond. Yeah, even in those 30% who responded, we could only reproduce half of the models. So half we could not reproduce. Sometimes they respond and they say, oh, right, my PhD student is not here anymore. I cannot find the code and so on. So we simply can't reproduce even after we contact them. Even sometimes they provide some information and that's not good enough for us to reproduce the model. Okay, moving on. So if you look at the age of the model, there's no indifference. This is the similar trend. If you can see the proportion of the model that are reproducible, not reproducible, more or the same across all the years that we have analyzed. And so you can see that only we could contact the authors in the last 10 years, not before. Okay. When we also looked at the journals and see if there's any clear pattern, if some journal is better, some journal is bad. We can't really conclude anything because few journals had lots of publication with mathematical models, and of course, a few journals had less models. So there's no clear pattern in terms of journals on the reproducibility of the model. We did compare the size of the model, the total number of species in the model, and what we found is that smaller models, of course, more reproducible, and bigger models are more, difficult to reproduce, but still a significant proportion of the small models are not reproducible as well. Okay. So the reasons why these models are not reproducible, we try to look into the reason. Of course, as we know about a lot of models were not reproducible due to missing parameters. They did not report a parameter value in the manuscript, and sometimes did not report initial condition. Sometimes we try to guess from the simulation figure. Of course, it's not possible to guess all the missing values for initial condition, and sometimes there is some error or in the equation of the model and so on. Okay, and for still a huge majority of the model, like 70 models, we cannot find any reason. There's no reason that's obvious to us, at least. So we try to explore, but there's no results. So that could be something wrong. It could be a wrong parameter, wrong initial condition, wrong equation. No one knows. Okay, so where are these models available? So all these models are now in Biomodels. So those which are reproduced or under the manually curated section of the Biomodels, and those models which are not curated, not reproducible, are under the non-curated sections of the Biomodels. So in the non-curated section, you cannot consider all models are bad, because this has models which are which fail to reproduce, and also those models we didn't have time to look at, and also those models or with the modeling formalism we are not yet curating. But we had under discussion to tag these models which failed as as failed models, or some sort of labeling, and we discussed with the community, and most of them gave us feedback that there should be some sort of label to indicate that those models which are failed should be distinctly labeled, and the reason for failure should be labeled. So we are in process of doing that. So hopefully we'll do that soon. Okay, so what is the solution? So we had proposed like an 8-point reproducibility scorecard, which can be used by authors or editors and reviewers to assess the reproducibility of the models. So these are the 8 questions we have here. So are the mathematical expression described in the manuscript, or are the parameters, initial conditions given in the manuscript? Are the changes in the simulation conditions are described in the manuscript? The normalization described in the manuscript? Are the model codes shared publicly? Are the codes available in a community standard format, or the codes in relevant database? Are there annotations or semantic enrichment in the model? Are the numerical results shared publicly in these models? So we came up with this 8-point scorecard to assess the reproducibility. We say that this can be used by authors, reviewers, editors to assess how reproducible a model is going to be. So is this a good indicator? So yes, we try to do some extra work. We picked up about 110 models and tried to calculate the reproducibility score for each and every model and ask if these are reproducible. So we set a cutoff. We propose a cutoff of 4 or more. Of course, not all models have all the tick all the criteria. So we found that if a model has got a score of 4 or more, it's highly likely to be reproducible. yeah, you can see that our odds ratio is 4.62, and from these 110 models which we analyze. So this is the solution which we think is proposed, and this is what we recommend modelers to use to assess reproducibility of the model. Okay, last slide now. So this work is now, I mean, it's been there out for a long time. It's in bioRxiv and is now accepted in molecular systems biology. Hopefully, it will be out there soon as well. It's quite tweeted and discussed in the community as well. And hopefully, there'll be some positive impact in the community, and there'll be more and more reproducible models in the field. All right. With this, I would like to end the discussion and acknowledge all the team members who to help to curate the models on the funding agency, which is IMI, and then BBSRC and the EMPL. And thank you for your attention. Of course, in the context of a subgroup, I'm co-leading the reproducibility group with Jakob. And if you're interested in reproducibility, I welcome you to join the subgroup. And I'm happy to take any questions.
Okay, thank you. So we have time. We have a strict cutoff at 4, so we don't have too much time. Reinhard, was there any business that we needed to urgently go through, or shall we have some time for some questions?
No, I think we have time for some questions. I think if there's time left, probably you should discuss your ideas about competitions.
Okay, so thank you to both of our speakers for very interesting talks. And I'd like to open the floor for 1 or 2 questions to our speakers.
So I have a question. Go ahead, Fred.
I was going to try to combine my 2 questions. I'd asked Kevin, thanks for that great talk, by the way, about stochasticity. And for the second talk, how do you deal with stochastic models and reproducibility?
Who's going first?
Kevin, I think you should go first.
Okay, I can go. I can say a little bit like the way we have some element of stochasticity rudimentally encoded in the very beginning is in the Poisson, like how many virions actually land on a cell. But beyond that, one of the points that we struggle with, which I glossed over, is if you have one virion that successfully docks and delivers into the cell, where does this continuum approximation hold water anymore to go on? And so I get in the inner workings, the way that we do this is we kind of run, we don't formally simulate the stochastics, but we don't read into any of those early simulations up until you get this critical threshold where what happens is that these RNA viruses steal membrane. And we basically right now arbitrarily set like 25 copies of this critical molecule that steals membrane. And then the architecture flips around and now the replication starts happening on a surface instead of in solution. And then that's where we say that there's an appropriate continuum regime approximation, and then we pay attention to those. But actually that switch is fascinating because we can only encode it a handful of ways to get all the nice calibration. There's other architectures that you think would work and it doesn't, it's much noisier. There's not anywhere near the reproducibility. And I actually think that might be saying something about what the membrane stealing might have an evolutionary advantage for this category of viruses. So I'll stop on that.
Okay, coming on to the next question, how are we dealing with stochastic models? So in this study, we just focused on ODE models for the sake of simplicity. And as I mentioned, so we do not want entire figure to exactly match, but as long as it reproduces simulation that doesn't change the meaning of the study, we accepted that as reproducible. So probably we apply the similar criteria there. So we do not expect one-to-one precise match with certain tau or precision here. Probably something similar will be applicable here.
So I have a question. First of all, there's great work for both of you. Thank you very much for both talks. Rahuman, so the results of your study are really depressing, right? So I'm wondering if so you developed these 8 rules that you postulate as a guide to reproducibility. Would you recommend that any journal that takes papers like this should enforce these rules?
Exactly, yeah, exactly. That's what we would recommend. So we kept it simple. There were other solutions. There were, some sort of additional curator-based evaluation of the models and so on. So, but we want to keep things very, very simple. So these points are very easy to evaluate. You can just have a checkbox and check them. And then, as I showed in the stats, then if the model has score of 4 or more, it's highly likely to be reproducible. So we will slowly start communicating with the journals and recommend that they use this this criteria to assess the models whenever there's a publication of model. Yeah, definitely, we will do that sooner.
Great, thanks. And just to follow up on Fred's question, you mentioned in the beginning that you also take things like agent-based models. What do you do with them?
Oh, so Biomodels as a database for sharing models, we consider all modeling formalism. So you can share and exchange models, but we haven't yet touched on agent-based models because we don't have expertise on that field. But then hopefully we will gain soon. So initially we have been only looking at OD models. Now we have expanded into constraint-based models. I'm in discussion with this working group, which is trying to develop standards to assess the reproducibility of the constraint-based models, and there's also this discussion of the logic-based models. So we are expanding slowly at the moment. So Biomodels has been initially known as a kinetic model repository, but now we will have soon curation done for these two, and hopefully in future we will also do assessment of agent-based models.
Great, thank you.
So Rahuman, John Rice, my concern is dissemination of useful knowledge coming from computational models to, let's say, a clinician. How do what is it I need to explain to clinicians about reproducibility as a characteristic of a model that they should be aware of and that would help them have some better trust or confidence that maybe a computational model just might be useful? How do we communicate that to people that we want to have using the output of models so that it becomes part of their knowledge of modeling and why they might be willing to accept models as useful?
I think, John, you asked a very good question. It's a very important question. I think there is no clear answer for that. But I think as in any experimental finding, reproducibility is very important, and it's important for us to convey. Instead of, for the clinicians, maybe first thing we need to convey is that we need to emphasize how important is or how useful are these models for them in their work or in their research and so on. So that could be the first step. And then the next step is, of course, making sure that they choose the models that are reproducible. So similar to the fact that they will prefer a study which is more reproducible for implementation or for any kind of clinical activities around that. So similarly, they should also pick up a reproducible model.
Yeah, but I think that at some point it's going to become very useful that people with your expertise and experience and like the examples that you were able to show could write the short kind of article paper that is written as though you were going to go in and teach a physician.
Yes.
That this information we're trying to give you came from a reproducible model. What does that mean to you? How do you explain that? I would love to see just a rough draft of a paper like that. And then we can figure out how we get it to people or make it better or whatever. Thank you. That's it. Yep.
Yeah, sure. Definitely. Thanks for the comment. I think I'll be interested. Yeah, definitely. This will be very useful.
I apologize. We're going to I don't want us to time out. Is there any more questions for Kevin quickly? We have time for one minute question. Jacob?
I have
1
one.
minute.
You use differential equations, and Fred was asking you about stochastic simulation. Did you try agent-based modeling, micro-simulation, any one of those techniques? Are they useful, and where do they fail if not? And I'll be quiet now.
Yeah, so shorter answer is we didn't, because it took us 5 years to get the continuum model to match to our experiments, which weren't single molecule. But now I think we can start looking into, I think what's clearly tricky is that at the end, you're dealing with millions and millions of individual molecules. And so that transition from the a discrete stochastic to continuum regime is a tricky one to sort of navigate. But we have a reference point now to feed that in.
Talk to me offline.
Oh, terrific. Excellent. Thank you.
So Kevin, John Rice again, I'm going to press my luck and give you the same question. It's like, I love what I think I understood you showed. What would that look like if it had no formulas in it? And you were setting out to go out and sell a physician on what you to show as a factor in how they treat patients.
Mm-hmm.
Could anybody write that lecture?
Yeah, well, and so I think that I was actually gonna weigh in on your prior question. Step one for one of the things that I think is a key aspect is user friendliness. I mean, the fear that most clinicians or basic scientists have in the command line is just that's a non-starter for people. And so for us, like this model is out on GitHub and things, and we basically just say, type this in and it will run.
Yeah.
README. And that's even not enough for virologists. And so it's next phase for us is I have a proper virology postdoc in my lab. We're going to do a graphical user interface where it's clicks and slider bars and just the graphs come up and go, because all of that makes it really reduces the activation energy barrier to adoption and experimentation and playing around with it to get some intuition about what it does and how to interpret it.
Yep. As this big working group forms up and these weekly meetings are phenomenal, that from the perspective of a dissemination supporting group within it, I would love to talk to you and a couple people offline on a Zoom about for demonstration purposes, what could we put together kind of quickly that we could actually take to a few MDs and sit down and say, try this. And because eventually,
John,
yep,
we're out of time.
I'm sorry. Okay.
But Kevin, if you want to put it up on a server to do exactly that, I can do it in an afternoon with you.
Fantastic.
It's
yeah.
Literally a 2-hour process.
Great. Yeah, like I said, I have a postdoc who's enthusiastic about these sort of things, and so absolutely. I mean, he knows how to do the model and everything.
Thank you, everyone. I know these talks excite enormous amounts of interest, and the format doesn't let us go through them. Please connect with each other offline, and I hope that these two very stimulating talks give rise to lots more. Again, our speakers today are welcome to come back and give follow-up talks. Look forward to seeing everybody again next week. We have a steering group tomorrow. So those of you who are on the steering committee know who you are. Please join us. And some of the material, the business material that we didn't get to talk to today, we'll talk to tomorrow. Thank you, everybody, for joining us. Thank you very much for our speakers and your patience in trying to squeeze things into a short amount of time. I apologize for not having more time for conversation and questions. Thanks, everybody.
Thanks very much. It was a lot of fun. Take care.
Bye.